Hi, I'm Ezgi. I'm a postdoctoral fellow at UT Southwestern Medical Center, Green Center for Systems Biology. Here I will talk about my recent work on non-canonical determinants of cell fate. A central question in biology is why do genetically identical cells behave differently under the same environment? In particular, what are the drivers of these different fates? And when do cells decide on a particular fate? To investigate these questions, I used budding yeast cells and their response to acute glucose removal. To this end, I grew them in a microfluidics chamber for 2 hours in rich media. Then for 10 hours, they were exposed to acute glucose removal. After that, the glucose was replenished again for 4 hours. When you follow the single cells, you see that upon glucose replenishment, some cells resume their mitotic cycles, like the orange and the green cells here. We call them quiescent cells. And some cells do not resume their mitotic cycles, and they are the senescent cells. To understand the difference between quiescent and senescent cells, I employed 5-color live cell imaging and followed cell cycle markers, stress response markers, lipid metabolism markers, interorganelle communication markers, etc. And I showed that these markers differ among the cells of different fates. Here I'm showing you an example, which is this marker for nucleus-vacuole junction expansion. You see that before acute glucose removal, the cells have similar amounts of this marker. However, during acute glucose removal, the marker diverges significantly among the cells of different fates. My next question was whether I can find a marker that is different even before acute glucose removal. And I found that the amount of the nutrient signaling kinase, RIM15, before acute glucose removal was predictive of the fate after acute glucose removal, which shows that stochastic variation in the nutrient sensing pathways before can set the cells for a particular cell fate. Next, using these markers and the Bayesian method of statistical evidence, I accurately predicted the fates of the cells before the glucose replenishment. So here I put an example of how you can employ this method. So here you see acute glucose removal 10 hours and the distribution of the marker we talked about, NVJ, nucleus-vacuole junction marker, for quiescent cells brown and for senescent cells blue. And here is the correct prediction percentage. So if at the beginning of acute glucose removal, these distributions are very similar and the percentage of correct predictions are about 50%, which is what random guess would do for a binary guess. But as these distributions diverge, the accurate prediction percentage also goes up, and using multiple markers, I was able to predict 88% of the cells, of the cell fates correctly. Next, using the same method, I calculated the probability of quiescence for every cell. Here I'm showing an example quiescent cell and an example senescent cell. What you see is at the beginning, the probability of quiescence fluctuates around 0.5. However, after a certain time point, which we call decision point, the cells settle on a particular fate. And I showed that cells decide on a fate within 3 to 4 hours after the initiation of the acute glucose removal. So thank you very much for listening to my talk. Please check out this paper, which will be published in Cell Reports next month and is also available on bioRxiv. Thank you again.